Welcome to part 2 of your brief tour of the cell. So I want to start off this web lecture by just doing a brief review of what we talked about in the last web lecture, specifically in the context of how all of these different parts of the cell help the cell to be the smallest unit that performs all of these functions we've been talking about that characterize things that are alive. So how can we think of a cell in and of itself, this tiny, tiny little package, as accomplishing all these tasks? And how do the different subcellular parts, how do the different parts of a cell help to contribute to all of these functions? And so let's just go through these. I'm going to add growth and development back into this list as I did when we talked about macromolecules. And homeostasis, obviously the cell membrane is hugely important for that. We bring that up almost every time we talk about the importance of the cell membrane. Everything that's alive has to have one. But also the nucleus containing the information to produce proteins, and the endomembrane system that's going to package those proteins and get them headed in the direction that they need to go are going to be important to maintaining these internal conditions within a narrow range that's optimal for life. So all of these systems are going to work together to maintain homeostasis. So we're not going to talk about gathering, transforming, and using energy from the environment at this point, because that's going to be the job of some of the structures we're going to talk about in this web lecture. But let's skip ahead to transmitting information through space and time. Reproduction, signaling, communication, obviously the nucleus is key to that. It stores all of that information that gets passed down from generation to generation, so the information through time. But also, again, the nucleus and endomembrane system are critical for producing the proteins that are going to be important in cell signaling, cell communication, the endomembrane system for packaging that information and sending it to where it needs to be used, whether that's outside the cell for communication with other cells or some kind of signaling within the cell. Responding to the environment, again, the nucleus and endomembrane system producing those proteins that are going to allow that to happen. Evolution, the nucleus is very important. The variability in individuals in a population is going to be the result of that information that's carried in the genetic material in the nucleus. Any changes to that genetic material, again, are going to be passed down through the generations, resulting in evolution. Growth and development, proteins, hugely important. So the nucleus, the endomembrane system, but also lysosomes as these centers for recycling of different materials in the cell. That's why we can make new proteins, new macromolecules, is because the lysosomes are breaking down those larger structures and making those raw materials available for further construction. And then also vacuoles, food vacuoles, these different little membrane-bound compartments are going to be important for growth and development. So bear this list in mind as we go through the different structures that we're going to be talking about today, and kind of tag them in your mind in terms of what kinds of processes such as these they might contribute to. So the first concept from your book for this web lecture is the idea that mitochondria and chloroplasts change energy from one form to another. So mitochondria are the sites of cellular respiration, which is the metabolic process that uses oxygen to generate ATP. So this process of converting carbohydrates to actual usable energy that the cell can use. Chloroplasts are found in plants and algae, and they're the sites of photosynthesis, so they're going to be harnessing energy from the sun to use water and carbon dioxide to produce carbohydrates that store that chemical energy that can then be metabolized and used to power all the functions of the cell. We saw peroxisomes in the last web lecture as part of the endomembrane system, because remember, they're important as membrane-bound organelles where detoxification happens and different oxidative reactions that need to be kept separate from the rest of the cell due to the toxic materials that are being processed inside. So mitochondria and chloroplasts have a lot of similarities with bacteria. Mitochondria and chloroplasts are both surrounded by a double membrane, and they contain free ribosomes and circular DNA molecules. So that's similar to prokaryotic cell, that circular DNA molecule. And the free ribosomes, remember, prokaryotes don't have that rough endoplasmic reticulum, and so they're just going to have free ribosomes. The same with mitochondria and chloroplasts. And they grow and reproduce somewhat independently in cells. They have their own processes for reproducing themselves and producing growth. And so these similarities lead scientists to believe that these organelles were once freely living bacteria that got engulfed by larger bacteria, and this led to what's called the endosymbiont theory. So let's take a look at how this works. Here we have the ancestor of eukaryotic cells with a cell nucleus, endomembrane system, and we can think of this as being the host cell. So at some point, this host cell would have engulfed bacterium that was capable of these metabolic processes that mitochondria do now. So an oxygen-using non-photosynthetic prokaryote, which for whatever reason did not get digested, was maintained within the cytoplasm and eventually became part of the cell instead of being an independently living organism. So they both got a mutual benefit. Probably the bacterium benefited from the protection of being inside this eukaryotic cell, and obviously the eukaryotic cell benefits from this oxygen-based aerobic metabolism, and eventually they became completely dependent on one another so that the bacteria actually became an organelle within the cell. Similar process suspected for chloroplasts. In this case, the bacterium that would have been engulfed, or the prokaryote that would have been engulfed, would have been a photosynthetic prokaryote. So remember last time I talked about how many prokaryotes have these folds, these membranous folds with chlorophyll in them that allow the prokaryote to undergo photosynthesis. So this first endosymbiotic event that led to the origin of mitochondria would have happened in the common ancestor of all eukaryotes. Remember, all eukaryotes have mitochondria. This second endosymbiotic event that involved the engulfing of a photosynthetic prokaryote would have happened in the common ancestor of green algae and green plants. So what I want you to notice is that this process of endocytosis, of taking in this bacterium by this pinching off of the cell membrane, accounts for the double membrane that surrounds both of these organelles. So remember, when things are taken into a cell by endocytosis, they are enclosed in a little vesicle of membrane. And so that's thought to explain the presence of a double membrane on both mitochondria and also chloroplasts. So let's take a closer look at mitochondria. We're not going to go into the details of how they do what they do. We're going to go into great detail on that when we get into cellular respiration. We're going to go over just the structure of mitochondria here. So mitochondria are found in nearly all eukaryotic cells, and they have a smooth outer membrane, but then that inner membrane has all of these membranous folds that fold toward the inside, the interior of the cell. And these are called cristae. And so these cristae are where many of the chemical reactions are going to be taking place. So the inner membrane creates two compartments. So the intermembrane space, which is between the outer and inner membranes, okay, so this space here, and then also the mitochondrial matrix, which is the space that is in between all of these little folds of membranous cristae, the mitochondrial matrix. So some of the metabolic steps of cellular respiration are catalyzed in the matrix, so just in this interior fluid in the mitochondrion. And then many of the reactions happen actually on these membranes. And so the cristae present a very large surface area with lots of embedded catalytic enzymes that synthesize ATP. Now let's take a look at chloroplasts. Again, we're going to go into detail on photosynthesis. We're just going to take a quick look at the structure of chloroplasts here. So most plant and algal cells have chloroplasts, and this is where photosynthesis takes place. Chloroplasts contain the green pigment chlorophyll. This is what makes green plants and green algae green, as well as enzymes and other molecules that function in photosynthesis. The chloroplasts have three membranes. The innermost membrane contains flattened sacs called thylakoids. So these sacs of membranes, they're often described as looking sort of like a stack of poker chips. And these stacks are called grana. Then surrounding the thylakoids is a space called the stroma. So this is just that interior fluid between these membranous sacs. So again, chloroplasts contain their own DNA and manufacture their own ribosomes, just like mitochondria do, which further supports that endosymbiont theory that these were once freely living, independent prokaryotes. So now let's shift gears a bit and think about the cytoskeleton. So the cytoskeleton is composed of these networks of long protein fibers that are organizing structures that give shape and coordinate the activities in the cell. So again, it's a network of protein fibers and it extends all throughout the cytoplasm. It organizes the cell structures and activities, it anchors the organelles and holds them into place. It is a dynamic structure. So this is not like the scaffolding that you would see at a construction site. These proteins move and change. They disassemble themselves in one place and assemble themselves in another. And this process of disassembling and assembling allows the cell to alter its shape in different ways. It can move things around within the cytoplasm, and it can even move the entire cell. So let's look at the functions of the cytoskeleton. So first of all, support and motility, the ability to move. So the cytoskeleton helps to support the cell and maintain its shape, and it's going to sort of provide what I like to think of as the highway system or the railroad tracks to interact with motor proteins to produce cell motility. So this is going to be kind of the rail that these motor proteins are going to interact with and kind of walk across. So inside a cell, these can be the routes by which vesicles travel to their intended destinations. So that's part of the answer to the question I posed at the end of the last web lecture. How do these vesicles get to particular locations? They are put onto particular pathways of microtubules that go right to where they need to go. And they're going to be transported by these motor proteins that are going to connect to the vesicle and also to this network of cytoskeletal elements. So I want to talk about three different kinds of cytoskeletal elements. The first are microtubules. So microtubules are the largest of the cytoskeletal elements. They're hollow rods about 25 nanometers in diameter and about 200 nanometers to 25 microns long, depending on the size and shape of the cell. Microtubules are constructed of dimers of a protein called tubulin. And so there's an alpha and beta unit to this tubulin dimer, and then these are going to polymerize together into this spiral shape to form a hollow tube. So functions of the microtubules: maintaining the cell's shape, guiding the movement of organelles. So if organelles are moving around through the cytoplasm, these are going to be the trackways along which they move. And they also are going to function in separating chromosomes from one another during cell division. So they're going to attach to those chromosomes and then disassemble themselves from one end and pull those chromosomes apart. Much more on that when we get to cell division. These originate from a place called the Microtubule Organizing Center, and there are going to be two distinct ends, a plus end and a minus end, and the plus end is where the rate of assembling these polymers is going to be greatest, and they're going to tend to grow outward toward that plus end. And they're going to radiate all throughout the cell. Remember, these are compression-resisting fibers that are going to maintain the cell shape and keep it from collapsing inward. So this microtubule organizing center in animals is called the centrosome. It's close to the nucleus. and it contains two bundles of microtubules called centrioles. So they're usually diagrammed looking sort of like short little pieces of licorice. They're composed of a set of tubulin triplets around a central cavity. So these are 9 triplets surrounding a central cavity, and the centrioles themselves are short little segments of microtubule, and they're just going to organize and send out this radiating array of new microtubules growing again toward their plus end. So the location is called the centrosome, and these structures that are organizing these radiating new microtubules are called centrioles. So microtubules are basically that highway system or those railroad tracks that serve for vesicle transport, taking these little vesicles from the endomembrane system and guiding them to where they need to go. So they provide stability, they provide a structural framework for organelles, and they're involved in movement. They're these railroad tracks. So vesicle transport requires a motor protein called a kinesin. So a kinesin uses ATP to do mechanical work. It's going to use ATP to create a conformational change in the kinesin protein. The head region is going to bind to a microtubule. So the head region is going to be on the trackway. The tail region binds to a transport vesicle, and it's going to kind of drag it along as the head region moves along the microtubule. So the kinesin is going to walk along the microtubule through a series of conformational changes as it hydrolyzes ATP. Every time it hydrolyzes an ATP molecule, it's going to have a conformational change that's going to cause one of these two foot-like structures to move forward and bind to the next active site on that microtubule. So we saw a short animation of this when we talked about proteins. Here's a sequence of images. So here is that tail region that's going to bond with the transport vesicle. There's a stalk, sort of the rope that's going to pull it along, and then the head is going to be composed of two components that are going to sequentially bond to the microtubule, hydrolyze an ATP. They're going to have a conformational change that's going to cause it to unbind and move to the next globular part of this protein as it sort of walks along the microtubule. So another structure that's formed by these microtubules are flagella and cilia, and these are involved in cell motility, so the ability of cells to move through their environment. So it's going to move the entire cell. So flagella are long hair-like projections from the cell surface that's going to be involved in moving the cells. Here's an image of a long flagellum coming off the end of a unicellular organism here. So there are going to be some differences between prokaryotic and eukaryotic flagella. So prokaryotic flagella are helical rods, so these spiral rods made of a protein called flagellin. And they're going to move the cell by rotating like a ship's propeller or like a wine corkscrew that's going to create a force pushing the cell forward. So in prokaryotes, the flagellum is not surrounded by the plasma membrane. It's going to be separate outside that plasma membrane. In eukaryotes, these flagella are going to consist of several microtubules, and they're going to have more of a whip-like action. They're going to flip back and forth, sort of like a fish's tail, propelling that cell forward. So you're probably familiar with this kind of movement in vertebrate sperm with their flagellum that moves the sperm. And in eukaryotes, the flagellum is surrounded by the plasma membrane. So given all of these differences in structure and material, it's thought that probably these two different kinds of flagella evolved independently from each other. So separate evolution of very different flagella in prokaryotes and eukaryotes. Cilia are structurally very similar to flagella, probably evolutionarily related to them. Rather than being sort of long and tail-like, these are short hair-like projections. Generally, whereas a cell would just have one or two flagella, a cell might have many, many cilia. And rather than rather than having this whip-like fishtail kind of motion, cilia beat back and forth more like a rowing kind of motion, so a power stroke, and then they kind of bend to come forward back in a recovery stroke, sort of paddle forward, kind of like with oars. So they have different kinds of movement, but they're the underlying organization of these microtubules that compose cilia and flagella. They have the same arrangement. So this is a complex 9+2 arrangement of microtubules. So we have 9 doublets of microtubules around the outside, and then a pair of microtubules that make up the center and act as a compression strut to maintain the length of that cilium or flagellum. So this complex, this 9+2 complex, is known as an axoneme. The axoneme is held together by connections called spokes. The ones going from the outer 9 doublets to the inner 2 are called spokes, just like the spokes of a wheel, and then there are links between them that hold all of this structure together. And then the axoneme attaches to the cell a structure called the basal body. So it's identical in structure with a centriole, meaning that it's got this outer ring of microtubule fibers, and this is going to be similar to the centriole. This is going to be kind of the center for organizing the growth of the axoneme. So what provides the force required for this movement? How does this bending of cilia and flagella occur? So this is going to be due to a motor protein called dynin. So the dynin forms a set of arms that create links between the doublets around the outside of the microtubule. So similar to the other motor proteins we've seen, it changes shape when ATP is hydrolyzed to walk up the microtubule. So this walking of one microtubule relative to the others toward the minus end, when this happens on just one side of the microtubule, it causes a shortening on one side. Those central 2 microtubules are going to maintain the overall length, and that's going to result in a bend in the flagellum or the cilium, which is going to allow this swimming motion. So now let's think about the second kind of cytoskeletal element, and these are microfilaments. So these are the smallest of the 3, and these are often called actin filaments or microfilaments, and they're formed by the polymerization of individual actin molecules. So these are little globular, spherical molecules that polymerize together to form long strands. So generally 2 strands of these polymers are going to coil around each other to form the actual filament. So these can be grouped together into long bundles, or they can be in mesh-like dense networks, and they form sort of a cortex just inside the plasma membrane. So this layer of this mesh of microfilaments is going to help to support and maintain the cell's shape. So this is going to give that compression resistance that will keep the cell from kind of collapsing and give the outside of the cell more of a gel-like consistency rather than the liquid consistency of the interior of the cell. So the functions of actin filament, as we just said, they help to define the cell's shape, and they can also produce movement along with the motor protein myosin. So same as the other motor proteins we've seen, it uses ATP to change shape and do work. So in the case of muscle contractions, this is an interaction between actin and myosin, where the myosin's head region is going to bind to binding sites on the actin filament and then change its shape. So it pushes the actin fiber in the particular direction, and lots of myosin heads doing this over and over and over again cause the shortening of muscles. They're also responsible for this constriction between the 2 parts of a cell during cell division that's going to tighten and tighten and tighten, and then eventually pinch off in the middle to form 2 cells. It's also responsible for this phenomenon called cytoplasmic streaming. So plant cells can be very large. They're occupied mostly by these large central vacuoles. And so in order to be able to effectively distribute nutrients and materials through the plant cell, these organelles are moved using these actin and myosin interactions with the organelles attached to actually keep these organelles moving and keep the cytoplasmic contents circulating to distribute materials around the cytoplasm. And also cell crawling is caused by actin filaments growing in one direction, moving the cells. So if we think of something like an amoeba, amoeboid movement, so this is going to be the result of these actin filaments assembling themselves on one side of the cell, disassembling themselves from another side of the cell, moving in this direction, and continually pushing out in this direction and constricting back in from the trailing edge, so that these structures called pseudopodia form and pull the cell in a single direction through this amoeboid movement. The third kind of cytoskeletal element are filaments called intermediate filaments. So intermediate filaments are basically defined by their size rather than anything consistent about their composition. And there are many types, each consisting of different proteins. So this is kind of a catch-all bin of all of these filaments that are smaller than microtubules and larger than these actin filaments. Along with these differences in structure, they're going to be diverse functions. So they're going to provide structural support for the cell. These intermediate filaments tend to be more permanent. They don't tend to assemble and disassemble themselves quite as much as the other kinds of cytoskeleton. And because of that, because they tend to be more permanent and stable, they're not going to be involved in movement. These filaments include about 20 different kinds of keratin. That's the protein that is synthesized in the epidermal cells of your skin. Keratin is what composes fingernails and hair and scales in reptiles, lots of different structures. And also that nuclear lamina, that interior structure underneath the nuclear envelope that maintains the shape of the nucleus, is composed of intermediate filaments. So it gives the nucleus its shape, it forms this mesh underneath the nuclear envelope. It anchors the chromosomes and keeps them in place. And these, of course, have to be more changeable than the other kinds of intermediate filaments that tend to be more permanent. These need to break up and reassemble every time the cells divide. So these are an exception to that rule that intermediate filaments tend to be a little bit more permanent. The last major concept in this chapter has to do with what's going on just outside the cell, outside the plasma membrane. And it turns out that what's happening just outside the plasma membrane is also very, very important to cellular function. So these extracellular components and connections between cells help to coordinate cellular activities and are, of course, extremely important when we start to think about multicellular organisms. So most cells are going to synthesize and secrete materials that are external to the plasma membrane. We saw that with the endomembrane system, with these vesicles being carried to the cell membrane and then dumping their contents to the exterior of the cell. So these extracellular materials and structures are involved in many cellular functions. So let's take a look at some of those. First one I want to talk about is the cell walls of plants. So the cell wall is an extracellular structure that's secreted by the cell, and that distinguishes plant cells from animal cells. Animal cells lack a cell wall. Prokaryotes, fungi, and some unicellular eukaryotes also have a cell wall. So the function of the cell wall is to protect the plant cell. It's going to maintain its shape. It's going to give it a rigid kind of exterior, kind of like a little exoskeleton for the plant cells. And it's going to regulate the uptake of water, so it's going to make sure that it's not taking up too much water. Plant cell walls are made of cellulose fibers. Remember, we took a look at those when we were talking about carbohydrates. And remember that they are these very stable networks of carbohydrate polymers that are joined together by hydrogen bonds to create a stable sort of meshwork of fibers. And these are going to be embedded in other polysaccharides and protein to give it additional cohesion and stability. So these can have multiple layers. So there's a primary cell wall that develops during the early development of a plant cell. This is relatively thin and flexible. There's a middle lamella, which is a thin layer between primary walls of adjacent cells. So some plant cells stay with just the primary cell wall and the middle lamella, but some other plants, as the cells start to mature, will secrete a secondary cell wall that's going to be added between the plasma membrane and the primary cell wall, and this is going to be a more rigid structure that gives more stability and more protection to the cell. So here's an image of these plant cells surrounded by their cell walls, and these plant cells are connected by structures called plasmodesmata. And so these are lined by the cell membranes of the different cells, and so this makes the cytoplasm of these adjacent cells essentially continuous. Materials can move between the cytoplasm of these different plant cells. And here you can visualize the primary cell wall, that middle lamella that's going to be between the primary cell walls of adjacent cells. So this is a separate cell here. And then this thick, more rigid secondary cell wall on the inside between the plasma membrane and the primary cell wall. So rather than a cell wall, animal cells have a structure called the extracellular matrix. So the extracellular matrix, like the cell wall, is secreted by the individual cells, and it's composed of glycoproteins such as collagen, proteoglycans, and fibronectin. Refer to your book for more information on those kinds of molecules. And the main functions of the extracellular matrix are things like stability, maintaining the position of the cell relative to other cells and relative to the body as a whole in a multicellular animal, and also integrating information from inside and outside the cell. So information that's coming from other nearby cells or from the environment is going to be integrated and that information is going to be brought into the cell through the elements of the extracellular matrix. So let's take a look at how this is arranged. So here we see collagen fiber. This is a very common structural protein in things like the dermis of the skin, and as an important component of bones and cartilage. And here is one of those fibronectin proteins that's kind of holding this cell in place and creating a communication between these embedded proteins in the cell membrane known as integrants. And so integrants refers to integrating. This is going to be where integration of information between outside the cell and inside the cell is going to take place. So signals are going to be communicated through the fibronectin, through these integrants, and into the cell. So ECM proteins bind to these receptor proteins in the plasma membrane called integrins. And this is going to have an important role in the lives of cells in terms of taking in information from the outside and responding to outside information. And the ECM can regulate cells' behavior by communicating with the cell through these integrins, and the result of this can be influencing the activity of the gene in the nucleus to change the proteins that are being produced in response to whatever signal is coming from outside the cell. So part of this environment is other cells, and in a multicellular organism, cells are going to be joined together, and they're going to be joined together in different ways depending on how those cells are interacting in the body. So neighboring cells and tissues, organs, or organ systems often adhere, interact, and communicate through direct physical contact with one another. So we already took a look at these plasmodesmata in plant cells that are channels that perforate the cell walls that allow communication between the cytoplasm of neighboring cells. Through these plasmodesmata, water and small solutes, sometimes proteins and RNA, can pass from cell to cell. In epithelial tissue, so what's an epithelium? It's basically a sheet of closely packed cells that are all in direct contact with one another. This is as compared to, say, like a connective tissue where the cells are kind of sparsely spaced within the extracellular matrix. So if we're thinking about an epithelium where all the cells are in contact with each other, we can have 3 different types of cell junctions. Tight junctions are basically seals between the 2 cells. They're completely sealed together so that extracellular fluid cannot leak in between them. So this is going to be an epithelium that's not going to let materials pass through it very easily. Desmosomes are anchoring junctions, and so you can think of these cells as being kind of riveted together in particular places, but still some material can pass in between those 2 cells, between these little rivets that hold them together. So they're fastened together in strong sheets, but still will permit some materials to flow through that sheet. Gap junctions are communicating junctions. These are similar to the plasmodesmata in plant cells that are going to provide cytoplasmic channels between the adjacent cells so that they can exchange materials through the cytoplasm. So here is an image of some of these different kinds of junctions. So we see tight junctions in this region where these cells are sealed together and no material can pass in between these. These are watertight, solid seals between adjacent cells. Here we see a desmosome, this distinct kind of riveted area where the cells are attached together. And then gap junctions between these 2 cells where there's an actual channel that goes between the cell membranes of the 2 cells and allows material to pass through. The last concept from your book is actually a nice one to end on because it kind of wraps all of this together into this very important concept of emergent properties. So the idea that a cell is greater than the sum of its parts, understanding what one component of the cell is doing doesn't really tell you what the cell as a whole is doing because all of these components work together to have properties that are different from the properties of the individual parts. So we saw this a lot in the endomembrane system, this very integrated system that works together to produce proteins that are going to go to specific places. The example from your book is a macrophage's ability to destroy bacteria, which is going to involve the plasma membrane to engulf that bacterium and take it into the cell. The cytoskeleton, which is going to bring it to specific places where there are lysosomes present. So that's how that vesicle that forms around the bacteria is going to end up moving toward a lysosome. And then the lysosome fusing with that vesicle to digest the bacterium and neutralize it. So think of these as integrated wholes. Always as you're learning this, concept maps are very, very useful here because it allows you to see how all of these different parts of the cell are going to interact to create cell function.